After layer is nutrient function. Sporogenous cell also divides results in formation of mass of cells, which is known as sporogenous tissue. This tissue is known as sporogenous tissue. Right? From this sporogenous cell, a mass of cells develop, which is known as sporogenous tissue. Development of pollen grains from this sporogenous tissue is known as microsporogenesis. Now we will study microsporogenesis. What is microsporogenesis? It is development of pollen grains from sporogenous tissue within pollen sac. Sporogenous tissue develops from sporogenous cell, right? These are layers, middle layer, septum, and epidermis, all these layers. This is sporogenous tissue developed from sporogenous cell by mitotic division develops a tissue which is known as sporogenous tissue. These cells within this tissue have connections with each other which are known as plasmodesmata connections. When they start maturing, these plasmodesmata connections get plugged by callose deposition. Each individual cell getting segregated from each other represents microsporocyte or microspore mother cells. These cells undergo meiosis. When each cell undergoes meiosis, they form 4 haploid cells. They result in the formation of each cell divides, resulting in the formation of 4 haploid cells. 4 haploid cells. Sometimes they are arranged in one is like this, second this, and third like this, 1, 2, 3, and fourth is behind. 3 cells are visible and fourth one is behind. That means tetrahedral structure. When you can only see 3 cells, fourth is behind, we call such type of 3-dimensional structure is known as tetrahedral structure. Sometimes after meiosis the cells form 4 haploid cells like this, which is known as linear tetrad. This is known as tetrahedral tetrad. You develop 4 cells like this. This is known as 4 haploid cells are formed, but they are isobilateral, and sometimes like this. If like this structure is developed from the meiosis, we call it a T-shaped tetrad. Sometimes it is like this. We call it a decussate tetrad. That means in simple terms, in microsporogenesis, sporogenous cell which mitotically divided result in formation of sporogenous tissue. That sporogenous tissue disintegrates into individual cells which are known as microsporocytes or microspore mother cells. Those microspore mother cells undergo meiosis, result in formation of tetrad. Those tetrads may be either in linear shape or be in tetrahedral shape, maybe isobilateral shape, maybe T-shaped or sometimes decussate. Within tetrads there are haploid cells. Those cells are actually polar. For understanding, this is linear tetrad consisting of 4 cells. They sometimes remain attached to each other even after the dehiscence, and sometimes separate prior to dehiscence within the pollen sac. That differs from species to species. But each individual cell, haploid cell, act as pollen grain. This is a linear tetrad. In each this tetrad there are 4 cells, 4 haploid cells. Each cell in this tetrad represents a pollen grain. This pollen grain consists of outer, at maturity there is an outer covering which is known as axene. inner covering, which is known as intine, and single haploid nuclei at the maturity divided into two cells, 1 and 2 cell, generative cell, and second is tube cell. Prior to dehiscence, each cell gets matured, result in formation outer axene, dentated axene, inner intine. Within intine there are two haploid cells. One is known as generative cell, second is known as tube cell. Right? That means in each tetrad there are 4 pollen grains. In some species, all the pollen grains within the pollen sac remains united with each other and unitedly. We call such type of structure as pollinia. Right? This is all about microsporogenesis. For development of this axene intine, there is one layer that is known as tapetum layer. This tapetum layer is known as nourishing layer. Tapetum provides enzymes, amino acids, hormones, and nutrients. Tapetum layer, which is interior layer, provides enzymes, amino acids, hormones, and nutrients to the growing sporogenous tissue for the development of these pollen grains. This tapetum layer consists of 2 types of cells. A is glandular tapetum. Second is amoeboid tapetum. And the cells within the tapetum are of 2 types. One is glandular tapetum, second is amoeboid tapetum. Glandular tapetum secretes different substances, but at the maturity they get disintegrated. In contrast, amoeboid tapetum is the tapetum in which cells merge with each other. That means the walls between them disintegrate and form a continuum amoeboid structure in which many nuclei, and this provides nutrition to growing pollen grains. Right? This glandular tapetum bears Ubish granules. Those Ubish granules secrete sporopollenin, which later on gets deposited in the axene of pollens. Axene of pollens for their strength. Everything which the sporogenous tissue needs for development of pollen grains is provided by this layer. This is known as tapetal layer. This was all about development of pollen sac. In next lecture we will discuss development of female gametophyte.